Welcome everybody to the 6th webinar of the online edition of the Protein Electrostatic Conference. I would like to remind all of you that we now have our program defined until the beginning of April, so you can have a look at the next talks. And next week, actually, there will be the first flash talk from Artemi Bendandi, who is a PhD student at IIT, followed by the presentation of Alexei Onufriev. The speaker of today is Frans Mulder from Aarhus University. Frans got his PhD degree from University of Utrecht in the Netherlands. Then he did some postdoctoral research at the University of Toronto and Lund. Then he was principal investigator in Groningen. And finally, in 2011, he joined Aarhus University as associate professor. His field of activities on NMR spectroscopy with main focus on structural biology and protein biophysics. Today, he will be talking about the accurate individual side chain titration curves measured by NMR spectroscopy. Thank everybody, and especially thank to Frans Mulder.
Thank you for selecting my topic for a presentation. Is it in the correct presenter view directly?
I would say so, yes.
Yes? Okay, great. And then I'll just minimize this screen to be able to see my own presentation better. Okay. Yeah, so what I'd like to do today is tell you about our efforts over some time in order to help understand electrostatic contributions to both protein stability, but generally also developing devising methods that allow us to get more accurate and reliable data that can be used to understand these systems, but also to provide benchmarks for computational methods in this way. And some of the tools that I'll talk about are also available from my website, the website of my group, which is protein-nmr.org. So what I'd like to go through today is first explain to everybody what the methodology is like that we have developed, and that some other groups are also still continuing to develop. Junji Iwahara and Flemming Hansen are also two key young people doing similar work at the moment. And the focus has been here all the time to be able to get access to individual titrating groups, which of course in proteins are the side chains, and to use that to do that by NMR spectroscopy, in order to get a faithful representation of the degree of protonation or the protonation state of these individual side chains. I will use one example, a small protein, the B1 domain of protein G, that we have worked extensively on in collaboration with the group of Sarah Linse. And here we have done a couple of things. We have devised NMR experiments that can specifically follow the titrations of the acidic side chains and the basic side chains in the folded state of the protein. By having those titration curves, we can then look into what are the origins of the pKa shifts. Why are some of them shifted up or down? What are the structural reasons, or what are the energetic interactions that cause these shifts? As I will show you, as we have the protonation affinity being changed during the pH titration, we actually get some very delicate information about the coupling and about the pH dependence of pKa values or proton affinity. And then I'll show you that we can determine these pKa's also in the unfolded state experimentally, but we also do this computationally nowadays. And by having access to both the pKa's in the folded state and in the unfolded state, we can also rationalize why proteins are display a certain stability profile over the pH range. And if there's time left, then I would also show a few things on photoactive yellow protein, which is a photoreceptor protein, which undergoes a photocycle, and this photocycle has a marked pH dependence. And there are some reasons why that happens, and we've been able to understand why it shows this pH-dependent recovery, which is related directly to the pKa constants of certain side chains in the two states that the protein has, in the dark state or in the light state, and specifically also some information about an arginine side chain, which was reported from other data to be neutral, and we decided to see if we could either validate or devalidate that for the system in solution. So of course, one can measure by various methods the net charge of a protein as a function of pH. And that is an interesting thing to do. But of course, you would know that this charge state of the protein is the result of many side chains and the termini of the protein itself that are being titrated in the experiment. Of course, we have a number of acidic side chains, aspartates, glutamates, tyrosine side chains, and the C-terminus. And then we have a bunch of basic side chains like the lysines, arginines, histidines, and the N-terminus. And as you see on this example graph, of course, the individual aspartates and glutamates might have quite different pKa values. They don't have all the same pKa value. But how can one get access to these titration curves for individual sites? That is the question here. So what we'd like to have is access to individual groups rather than the overall behavior of the system, meaning that we have access to the microscopic states. And the nice thing about the NMR methodology is that these probes are non-invasive, so we don't really need to change our system, except for the fact that we produce our proteins with the incorporation of isotope labels in order to get a high abundance of the NMR active form of those nuclei. So for carbons, we'll resort to carbon 13, and for nitrogen, we'll resort to nitrogen 15. The protons are already, of course, at 100% natural abundance or close there too. So here's an example of what happens when you have a small molecule that has a number of titratable groups. You see on the lower corner here the structure of the amino acid histidine. It contains 3 titrating groups. It contains the amino group, the carboxylate group, and the side chain. And of course, at very low pH, or in this case PD, because the experiments were done in D2O, of course, all these groups are protonated, and we get an NMR spectrum. And in this NMR spectrum, the signals come from the 2 protons that are bound to the carbons on the side chain ring. So that's the C2 and the C4 here. Those are those 2 proton positions. And then we have the alpha hydrogen at this location and the beta hydrogens at this location in the molecule. Now, when we start changing the pH, you see that the chemical shifts, they start to change. So the position of our resonances in the NMR spectrum, they move in response to the changes of the charges being developed or being removed during the titration. What you also see is that they move in quite different ways. And it's much easier to plot this, of course, by taking the position of such a resonance line as a function of pH in order to generate a titration curve. And those would then look like this. So for example, if one follows the titration curve, so the chemical shift of the resonance on the ring, then one sees a 2-step titration with a midpoint somewhere around close to 6, which of course coincides with the pKa value of the histidine side chain ring, of the imidazole ring. Whereas if you follow the hydrogen alpha, you see that there's a titration here at high pH around 9, and then there's another titration again somewhere around 2. And of course, those are due to the carboxylate titration at around pH 2, and the titration of the amino group here at high pH. So depending on where we observe in our NMR spectrum, which nucleus we use to observe, we actually get different information about the different groups that are being titrated. Now, the problem is that all of this was initially developed by proton NMR. And proton NMR has the advantage that it is very sensitive, but it does have another disadvantage, which is that protons are very sensitive to many things. And I often show this slide to the students where I say, okay, we have aspartate 26 in this protein. If I'm not mistaken, this was thyroidoxin. This is work from Julie Foreman-Kay from the last millennium. And she did titration of this protein and followed a number of atoms or nuclei of this protein in the different side chains, which she had all assigned. And then you get a response of the alpha hydrogen of the aspartate, as in the top graph, and the two beta hydrogens that sit next to it show the behavior in the middle and in the lower panel. And then I always ask, can anyone tell me what is the pKa of aspartate 26? And then it's silent for a while, like now. And then some people have a guess. But the problem is with this kind of data, even if you have a guess, you're never quite sure. So the problem is that these protons, they respond to everything. They respond to the changes in the electron densities of the side chain of this aspartate, but they also respond very sensitively to charges that are developing in their environment. And the problem is that if you have this mixture of effects, you don't really know what you're looking at. And so in order to get away from this, we decided that what one should really do is look at nuclei that are as close as possible to where the protonation or deprotonation event takes place. So for example, in the case of aspartate residues, it would be the best if we could follow this gamma carbon of the carboxylate group, because this is going to be dominantly changing its chemical shift because of the electron density of this group changing rather than things happening in the environment. So the approach we took is try to develop NMR methods that allow us to follow these chemical shifts that are going to be uniquely sensitive to the protonation of a certain side chain. The added benefit of going to carbon NMR is that we can do 2D NMR spectroscopy such that it's easier to resolve the individual resonances once we have made an assignment of the peaks in our spectra. And I'll show that in a second. So we spent some time trying to figure out, okay, if we want to do this, if we develop these methods with carbon NMR or nitrogen NMR for all the different side chains, which should be our preferred probes that we should be following. And there's a lot of literature data on this, and we did some things ourselves as well. For example, for the aspartic acid, it is best to take the carboxylate carbon, the same for the glutamic acid. For the tyrosine, one actually has two choices. We could try to address the carbon zeta, that's the one where that's going to be deprotonated, the oxygen is going to be deprotonated. But it turns out that if you look at the carbon gamma at the other end of the ring, then that is just as sensitive. And there is some NMR, well, there's some reasons that have to do with the flipping of the tyrosine rings, the rotation of the rings about the twofold axis that causes the gamma shifts to be easier accessible spectroscopically than the zeta shifts. So that's why we developed some experiments following the C gamma shifts of the tyrosines that was published some years ago. For the basic side chains, we follow this a similar rationale, so get as close as possible to where the action is. And that means for lysines, one follows the N zeta here. For histidines, one can follow the nitrogens in the ring. Those have huge shifts. And for arginines, that turned out to be quite challenging. And there have been also some beautiful work from Fleming Hansen's group recently to address this. And I'll show you a little bit at the end of how arginine can actually be useful, or how one can do the arginine experiments following these nitrogens, despite the fact that also the arginine guanidino group is undergoing rotational motion, which causes some problems in some of the NMR spectra. But one can get around that nowadays by using carbon observation NMR. So if one is interested in more details about this, then I would refer you to a review that we've written a few years ago, where we have collected all this data about all the possible NMR experiments that one might want to do. Then of course one has to a choice of labeling, and one can either try to look at only a certain type of amino acids. This has been an approach that was taken also, let's say, in the last millennium. So for example, one could take lysine residues that are selectively labeled with a C13 disposition in the side chain. And then one has a relatively simple spectrum because it only contains as many peaks as there are lysines. Nothing else is labeled here. For aspartates, one could try to get labels in this position, and for glutamate in this position. That has been published work also by Sarah Linz's group. This is mostly quite more a little bit more difficult because aspartate and glutamate are very central to the TCA cycle, so that if you put these labels in, they easily get scrambled. That's not the case for the lysine. We decided to go for uniform C13 labeling, so everything is going to be labeled in our samples, and then we have to rely on us being NMR spectroscopists in order to devise smart enough experiments such that we can resolve these individual groups spectroscopically, even though there might be many. And that is not, that is a surmountable task. So here I give you an example of what kind of experiment one could then imagine doing. An NMR experiment always is pulsing on certain nuclei, transferring magnetization from one nucleus to another nucleus, letting that evolve for some time so that we observe the chemical shift of that nucleus, and then returning to usually protons in order to do the detection. So in this NMR experiment that is called HC bracket CO, it means that we start on protons, in this case the beta protons, that the magnetization is transferred to the C beta carbon. This is between brackets, meaning that we're not observing this atom. And then we transfer it on from this carbon beta to the gamma carbon. We let the magnetization evolve for a while so that it encodes the chemical shift of this side chain carboxylate carbon. And then we return to the C beta and we return to the H betas, and we observe the signal by detection on proton. That gives these kind of spectra for a small protein. This is PGB1 at close to neutral pH. So each side chain, aspartate, asparagine, glutamate, and glutamine, will contribute signal in this area of the spectrum, in this HCCO spectrum. So you see along the horizontal axis, we have the hydrogen chemical shift. Along the y-axis, we have the carboxylate carbon chemical shifts. And of course, since each aspartate or glutamate has two hydrogens, you see that there are two peaks at the same carbon frequency for all the side chains. Sometimes the two hydrogen frequencies might be coinciding, such as in this situation here. In this spectrum, you also get some bonus material, although of course the backbone carbonyls are not going to be titrating. We have observed that some backbone carbonyls actually do show titration curves, and those titration curves are due to the fact that they are involved in hydrogen bonds. So the carboxylate carbons or the carbonyl carbons can sometimes act as acceptors for hydrogen bonds of side chains, and so that one can determine the side chain pKa by the backbone chemical shift of a carbonyl, and then one can directly observe that there's a hydrogen bond interaction between a backbone and a side chain. So here's then an example of using this experiment, this HCCO NMR experiment during a titration. And you see here a number of pH values that have been colored differently. So it's the same spectrum as before. We start at high pH values, then we get one NMR spectrum, then we change the pH of our sample, we record the next two-dimensional spectrum, et cetera, et cetera, et cetera. And what you can see here is that despite the fact that the hydrogen chemical shifts of this aspartate here doesn't really change all that much, the carbon chemical shift undergoes a huge change. So the carbon chemical shift here is now what we're going to be plotting as a function of the pH, and that is going to give us a titration curve, and that is an individual titration curve for this particular aspartate residue, and we'll have another titration curve for this aspartate residue or glutamate residue, and so forth. The data then looks like this. So this is experimental data. The data points are shown here as circles, so they're really, really tiny. And because it's an NMR chemical shift that we're observing, the data is also very highly precise. And I show you here all the glutamate residues of this little protein, observed between pH just below 2 up to almost 10. And we've done these experiments at 2 salt concentrations, which will become apparent why we did that. And you'll see in light blue that you get a really beautiful titration curve for each glutamate, individual glutamate residue, but that the appearance of each curve can be quite different, and also that the pKa constant can be quite different for some residues. So in the values here are the pKa constants, so some are upshifted and some are downshifted, and obviously the reasons for those are that there is either barial or some electrostatic interaction or hydrogen bonding. Here is an example from the same experiment. In the same experiment, we observed the glutamates. We also observed the aspartates. So this is experimental data. The model pKa is around 4 for aspartate. And you see that some of these residues have a really downshifted pKa value. So they really like to become deprotonated, whereas some have a very highly upshifted pKa constant. We did the experiment, as I said, also at a higher salt concentration, and you see that these curves, they do move. So apparently there is some electrostatic screening that can be removed by the addition of salt. But in some cases, this is also quite small, this contribution. So there must be other origins that are responsible for the pKa being so shifted. A typical way of analyzing such data is the Henderson-Hasselbalch equation. So this is an observation that relates the observed chemical shift as a function of the pH. And that, of course, gives you then the limiting values at the ends for the protonated version and the deprotonated version. It gives you the pKa constant, and it gives you a parameter called the Hill parameter, which is an empirical parameter that is giving information about the slewness of this curve. So whether this curve it doesn't have a steep appearance, but a rather shallow appearance. And of course, in electrostatics, that is always due to negative cooperativity. What we have done, instead of looking at the titration curves, is actually to look at the derivatives, because that is much more telling. So if you look at the derivative, and this we call binding capacitance curves, if one has a model pKa value that doesn't, so a titrating group that doesn't interact with anything else, then you expect to see this light blue curve here. So the first thing you notice that this is symmetric, and the maximum is at 0.58. What I show in the same plot here for glutamate 56 is the experimental data in purple, and you see that this is far away from having a maximum at 0.58, but it's also very far away from being symmetric. So if one does this Henderson-Hasselbalch fitting with this Hill parameter, one assumes that the pKa constant doesn't change with the pH, and therefore this curve is symmetric. It doesn't fit, however, with the experimental data. And the same thing happens here with glutamate number 8. So also here you see that the curve is much shallower, the titration curve, meaning that the derivative is much lower, but you also see that it's far from being symmetric. Here it seems to be behaving with a constant pKa value, but the proton affinity really changes here, meaning that the electric field that it senses is really changing over this pH range. The reason for this obviously being that there are other acidic side chains that are titrating, and so charges are being formed as we increase the pH. I think one of the most telling results that we got from this analysis were these curves. So if we have an aspartate that is the first one to titrate, then it shows an almost ideal curve, because it doesn't really notice yet that there are other titrating groups in the protein here. So there's no charge-charge interactions. The curve is symmetric, and it has a maximum very close to 0.50 or to 0.58. The same for those residues that are very highly upshifted. They also titrate in isolation, so to speak, in a constant field. The field doesn't change as you change the pH. But in other curves here that are slightly downshifted, or ones that are slightly upshifted, they have this trailing behavior at one end as multiple groups are starting to deprotonate here. So this is a very nice demonstration, I think, experimentally. So this is experimental data in purple here, that you see these asymmetric curves that cannot be understood simply by using a Henderson-Hasselbalch equation with this Hill parameter. We really have to consider now that the pK constants are pH sensitive. They're really changing. So one thing that we then did is obviously the experiment with removing the salt or adding the salt to see if one can screen out these electrostatic interactions. And if you look at the derivatives, I think it's pretty easy to see that these curves, they do move all closer to the model pKa value, and they also become, they also increase their amplitude. So very clearly, charge-charge interactions play a role in both the magnitude of the pKa shift and the asymmetry of the behavior here. As I said, in the same spectrum, we do also get our carbonyl shifts. And for example, lysine 10, this is the carbonyl group that we see of lysine 10. It shows what we call a ghost titration. Of course, the residue itself cannot titrate. It's a backbone carbonyl, but you see that it responds to something else that's happening close to it, which is actually a hydrogen bond interaction with a side chain of glutamate 56. And you see that these two curves also give the same pK constant, which is the one of the side chain of glutamate 56. So there's extra information in these spectra. So these are the three causes of pKa shifts in proteins: beryllium in low dielectric environment, the hydrogen bond formation in either the protonated or deprotonated state, and then the charge-charge interactions. So one thing that we tried to do now is that now that we have access to all these individual titration curves, then we figured it should be possible, given alt literature, to understand also the pH-dependent stability of the protein once we have access to all the pKa shifts inside the folded structure. But of course, we should then also know what the pKa's are in the unfolded state. So therefore, we continued our effort and also tried to develop a few methods to determine pKa's in the unfolded state. And we did that in this case by making fragments. So we took PGB1 and cut it into 2 fragments. In this case, the protein no longer folds. The 2 fragments themselves are not enough to make a stable structure. And then you see here the same kind of experiments as I showed you before, these HCCO experiments where we follow the carboxylate carbons as a function of pH. And so now we also have access to the pKa constants or the entire titration curves of the unfolded state, and we can use those together. So again, I'd like to draw your attention to these proton binding capacitance curves, because I think they are rather illustrative, saying that, for example, aspartate 36 has pretty much the same behavior whether it is in the folded or in the unfolded state. Whereas for some residues like glutamate 56 or aspartate number 8, in the folded state, they're very, very different. So there's interactions in the folded state that are responsible for this very clear difference in their titration behavior.
Frans, may I have a question?
Yes.
Are these kind of measures time resolved? So can be done with some, can one monitor the evolution in time of the states? Or if so, with frequency?
So each of these 2-dimensional spectra takes maybe 20 minutes to record. So if you'd like to get this wealth of data, it really can only be used for things that would change very slowly in time. Yeah, it might be very challenging to do the experiments in different ways that would give you a more reasonable time resolution than minutes. But then collectively having the access to the exact protonation state at each pH value for each residue inside the protein, we could measure the denaturation as a function of pH by fluorescence methods. And then we could take our pH-dependent stabilities that we calculated from the pKa values. And you see that this actually can represent the experimental data quite well. And so explaining also why proteins, of course, are mostly stable at certain regions of pH. If one does not have access to the unfolded state, or if the unfolded state contains just too many residues that they're hard to resolve spectroscopically, the alternative is to do this by calculation. Here we followed a recipe that was presented by Professor A.A. already many years ago, And we have changed the approach that was taken. So we've taken the calculation methods that was presented in this paper, which is based on the Debye-Hückel theory, coupled with Gaussian chain statistics. But rather than using a Monte Carlo approach, we've used a hybrid mean field approach that was published a few years ago. And this program is available if you'd like to play with it. The nice thing about going away from the Monte Carlo approach and going to a hybrid mean field approach was that this was necessary for us in order to work with IDP, so intrinsically disordered proteins, because in that case, you don't have 6 or 7 titratable residues, but you have 40, and then the Monte Carlo approach is obviously going to explode. And it's also not necessary because many charges are far removed from each other, and thereby it is also suitable to a mean field approximation. The next topic that I want to cover in the final 10 minutes is, what if we cannot titrate the protein? So if we cannot do a titration experiment, is it still possible for us to determine what the protonation state actually is? And as I showed you, the way that we do the titration experiments is that we follow a certain chemical shift, and we follow it from low pH to high pH. And then of course we know if we see a transition, we know of course what the protonation state is at a certain pH value. The problem is, if we don't see a titration, what is then the protonation state? Can one just use the NMR chemical shift to say, well, this one is protonated or this one is protonated? I would say no. Especially if residues are buried inside a low dielectric environment, chemical shifts can be quite significantly moved, making it very dangerous to assume, based just on an NMR chemical shift, whether a group is protonated or deprotonated. In that case, the best way is actually to see if one can observe the hydrogens that are involved in the form that is protonated. And to show this I want to illustrate this with one example, which is the photoactive yellow protein from a small halophilic bacterium. And this is a small protein that has a structure in the dark state that, when illuminated, it contains a chromophoric group. In this case, this is a parachimeric acid chromophore. Once illuminated, it very rapidly undergoes a number of changes. Those have been, of course, characterized by optical methods, time-resolved optical methods. And then it ends up in this PB state, which is very blue shifted. That's why it's called PB. So PG is a yellow shifted or green shifted version, and this is a blue shifted one. And this blue shifted form has now a different protonation state of the chromophore, but also other residues in its environment have changed protonation state. And that is reflected by this recovery curve. So the recovery of the blue shifted intermediate back to the ground state is very highly pH sensitive. So it has an optimum around pH 8, which is where it is fastest. But if you go to a either very high pH or very low pH, then it becomes very, very slow. And the question was, why is this? And can we now use these methods that we've developed to determine all the protonation states around the active site and maybe find the explanation here? The other thing that was that happened was that groups in Japan used a combination of X-ray crystallography and neutron diffraction in order to determine the structure of the PG state, in which they found that the deuterons were actually very closely located at the middle of a side chain of glutamate 46 and the parachimeric acid chromophore. So that is illustrated here on the right. So the parachimeric acid chromophore was suggested originally to be negatively charged and then forming a hydrogen bond with a protonated glutamate. And then an arginine was very close by and stabilized the negative charge on the parachimeric acid by being obviously protonated. But from this work, the suggestion was then that if this hydrogen is very closely in the middle of these two oxygen atoms, it might constitute a so-called low barrier hydrogen bond. And the other thing, what they found based on the density of the neutron structure, was that the arginine was then expected to be neutral, which was a very surprising result, meaning that its pKa must have been shifted by several units from its normal pKa constant, which is actually higher than 13 in solution. So we looked at this by NMR spectroscopy. Here is our NMR tube with the protein. It's nice and yellow. And also Dan Herschlag's lab has done very nice work, also reconstituting the protein with different forms of the chromophore in a similar manner as what was presented by Professor Boxer a few weeks ago, being able to add small modifications to the parachimeric acid chromophore rather than having it reconstituted from. Well, if you reconstitute it, you can change this chromophore by putting halogens on these side chains, and thereby you can change the actual electronic environment here, and also the pKa constant of this group here. So the nice thing about this is that in a simple one-dimensional proton NMR spectrum here, you can see these two peaks, and these two peaks have been assigned. One of them is then associated to the hydrogen drawn in red here that is associated with the tyrosine, and the other one is the hydrogen that is observed between the parachimeric acid chromophore and the glutamate residue. And what we did is we then tried to determine all the groups that are located around this parachimeric chromophore. The parachimeric acid itself is not isotopically labeled in the protein that we produce, so only the side chains of the other groups can be observed here. So we developed some new experiments to follow the tyrosine, the glutamate, and also then the arginine to see if in fact it was neutral. And here are the UV absorption spectra that you get if you illuminate the protein, where the PG state is being reduced and the PB state is being increased if you do these experiments in an NMR tube, which we did already in the 90s. So the explanation finally for why we observe a maximum in the recovery around pH 8 actually comes from the fact that it does not come from the fact that the protein itself has any groups that are titrating in these areas. That is not happening at all. So we followed all these hydrogens that are in the active site here, and none of them change between pH 3 and 11. So they're always the same. They're always in the location where they are at pH 3 as they are in pH 11. But what happens is that a hydrogen that is in the ground state, this hydrogen is bound to the glutamate, and so that this pKa is very high, whereas the chimeric acid, which normally has a pK around 8, this one is much reduced, so that this one is deprotonated. So in the PB form, this group is going to be protonated and the glutamate is going to be deprotonated. And what needs to happen is a simultaneous thing that both E46 needs to be protonated to return to the ground state, but the PCA needs to be deprotonated. And the maximum occurrence is, of course, the product of these two curves, which has a maximum in the middle. So that is what is happening. So that explains the pH dependence of the recovery. It's just due to the pKa, the differences in the pKa constants in the ground, in the folded state here and in this PB intermediate. But then this leaves the question of arginine 52 that I just want to use the last few minutes on. As I said, the pK of arginine is very high. It's around 13.8, as was studied by Lawrence McIntosh by NMR spectroscopy following certain probes in the side chain. So in this case, they chose the 13C zeta carbon in the arginine and showed that you can really make this change by about 4 ppm if you go to very high pH values. So we tried to see if this group would titrate in our protein. So we recorded the carbon NMR chemical shift at many pH values, and you see that the peak, there's a single peak here, and it never changes. So whatever the protonation state of the arginine is, it doesn't change. That means that we still don't know what the protonation state actually is, and for that we had to actually develop a new NMR experiment. And in this case, rather than doing proton-carbon or proton-nitrogen correlated 2D NMR, we did carbon-nitrogen 2D correlated NMR, because in such a spectrum where we actually observe the carbons, one can find correlations between carbon and the N-epsilon and the N-eta groups. And it's of course the N-eta groups that are going to be protonated or deprotonated in the titration. So if we can follow those nitrogen chemical shifts, they should change by more than 10 ppm. So the NMR experiment, which I will not show, but it's published material, starts by taking the magnetization from the protons, then transferring that by cross-polarization step to the nitrogens, which chemical shifts we then measure, and then we transfer on to the carbons where we do our detection. And that then gives us these two maps. Now, in the case of photoactive yellow protein, there are only two arginines in the protein, arginine 124 and arginine 52. And arginine 124 is typical of a surface exposed arginine. It rotates very rapidly around this NC partial double bond, causing the nitrogen chemical shifts of the two sides of the arginine side chain to be identical. So we get an averaged location for that particular nitrogen frequency. However, arginine 52 is obviously rotating very slowly, and that slowly in this sense means on the order of milliseconds or slower, and causing the nitrogen chemical shifts of this group and of this group to be different. Now, of course, now we only have the chemical shifts of these two groups. We still don't really know whether it's protonated or deprotonated. And for that, we use a simple trick, which is that we can also in NMR, besides NMR chemical shifts, we can also observe NMR spin-spin splittings. So if the nitrogen would have a single proton associated to it, bound to it, then it would show a doublet. And this is the case for the N-epsilon position. So you see doublets, very clear doublets, split by close to 90 hertz. However, if you go to the arginine side chain of the eta groups, then you really see that they're triplets. And it's a triplet here and it's a triplet here. And the triplets arise with a 1-to-1 intensity ratio because of the observation of two nitrogen proton splittings on top of each other. So this directly demonstrates that this peak and this peak, they both arise from NH2 groups, meaning that the arginine 52 in solution is actually charged. So with that, I'm at the conclusion of my story here. What I hope I have conveyed is that NMR chemical shifts are very highly sensitive and very faithful reporters of changes in electrostatics, and that we have now developed a whole suite of NMR experiments that depend on using carbon-13 and nitrogen-15 reporter nuclei at individual side chain positions in order to follow titration behavior. And we can follow now, we have these experiments are now available for all possible side chains, tyrosines, cysteines, etc. The only thing that is, of course, important to realize is that the NMR experiments are very sensitive for small proteins, and as proteins get larger, then this poses a problem, or one should revisit this and actually develop NMR experiments that are also suitable for large proteins, which can nowadays be done with carbon observation and in combination with deuteration. So there is actually work to be done if people are interested to continue this kind of work. And we think that the examples that we have shown and that many others have by now demonstrated, and I'm sure that also Junji Iwahara will show us some very nice results in a few weeks, really allow us to differentiate between sophisticated or more sophisticated electrostatic models than just the Henderson-Hasselbalch equation, and really allow us to sense the pKa constants of proteins in solution and provide in that way also good benchmarks for computational methods. And then finally, spin-spin coupling patterns allow us also to determine protonation states in the absence of an actual titration. And with that, I'd like to thank many people who have been involved in this work over the year, collaborations with Professors Kataoka and Kamekubo at NICE in Japan and Sarah Lins at Lund University, and my former colleague Rutsek at the University of Groningen in the Netherlands, and all other PhD students and postdocs. So with that, I'd like to thank you also for your attention.
Thank you, Frans, for your very interesting presentation. I see that Stephen has already a question for you. Go ahead.
Yeah.
Yes, hi. This was a really nice overview of this area. I really appreciate it. I'm going to make sure that everyone in my lab watches this. I just wanted to ask a technical question, something you mentioned early on but never revisited, and that is the business of tyrosine labeling and where you locate that. We've as you may know, we've done a lot with C13 labeling at the carbon adjacent to the hydroxyl group in KSI and GFP, even in reaction centers in very diverse environments. It's super sensitive. But you mentioned that there was a problem, and I'm wondering if you could sort of discuss what that problem is and why we shouldn't be doing that.
Well, it's an NMR specific problem,
Okay.
and it has to do with ring flips. So maybe it's good if I can go back to the slide. I think I can even do this on my bar here. Right. Yeah. Wow.
Yeah.
Beautiful.
Yeah,
I've never used this feature, but here. Oh, no, sorry. It was right here. Yeah. So in order to get resolved two-dimensional spectra, we need to have some form of 2D NMR. And the most sensitive ways of doing 2D NMR is connecting a proton chemical shift to this carbon chemical shift in the ring. Now, if one wants to observe this one, this is the one that actually will change the most. So this is the ideal one to be looking at. The proton that you have to correlate it to are these epsilon protons. However, what happens if this ring starts flipping, then these chemical shifts are going to be averaging. And if this process happens to be on a millisecond timescale, which is quite common for tyrosine rings, this broadens these signals completely away. So when we applied this to photoactive yellow protein, we could only observe a small subset of the tyrosine residues. So what we then resorted to was actually taking the gamma chemical shift and correlating that with the beta protons, because the beta proton chemical shifts and the gamma carbons, they're immune to this rotation. So in such an experiment, and then we observed all the correlations.
I see. Okay, so it is a very NMR specific. I guess in our case, we were just lucky. We could directly see that carbon, and it is super sensitive.
But if you do one-dimensional carbon NMR, it's fine. So because also this carbon is immune to the flipping.
Exactly. Yeah. There is the subtle issue of the orientation of the proton on the hydroxyl group, actually, and that can have interesting effects. It's been very little studied as far as I know, but
I think Kainosho has done something on this. Takeda and Kainosho have looked at this. Yeah,
it's difficult, and there's a little bit of neutron diffraction data that's been done, because it really matters if you're interested in the electrostatic interaction with something nearby. Then the orientation of that OH, that OH dipole can actually matter a lot. That's a topic that Marilyn will be familiar with. That came up extensively in simulations from Bill Parson and Ariel Warshall in the Reaction Center. Anyway, thank you for your comment. That helps.
There is a question from Jim Warwicker.
Hi, thanks, Walter.
Hi.
Thanks, Frans. Very, very interesting and precise, much more precise than some of it, or me anyway. Question about the unfolded form, actually, because I mean, I'm sure many of us remember sort of Oliveberg and Furst and discovering, pKa changes at 0.3 or something. I'm wondering if you're seeing any, and when I looked at it, there was some data from Fink, I think, in about 2000 with different folding energies, and you could benchmark in a much rougher way than you've been doing with calculations. But I'm wondering whether you see, how much does the pK changes you get in the unfolded form ionizable groups, how much does clustering of those groups matter? And then I see that you're interested in IBRs in general from literature, and I'm wondering about obviously that LLPS and such like, and how do you see that area?
Yeah. Oh, that's a very good question. And we have some more recent data that I decided not to include yet in the presentation, because I'm also still hoping that someday we'll have the real physical event, and then maybe I'll be allowed to come and present some of that work as well. But so what we observed in an IDP like alpha-synuclein, for example, is the fact that that the pKa's of aspartate and glutamate residues, that they are moved by about 0.4 units, pKa units, is very common. And of course, in regions that consist of alternating charge, then you see that the pKa's are downshifted because they have a favorable interaction with the lysines that are in those regions. Whereas if you go to the acidic tail of alpha-synuclein, then all the aspartate and glutamate pKa values, they're upshifted by about 0.4 units. And that was something that was experimentally looked at by Alexandrescu's group. And in fact, in the bioinformatics paper, we used their data to compare how this hybrid mean field method would compare with their experimental observations. And we found, at least qualitatively, that the order of magnitude agrees quite well. So the model, the simple Debye-Huckel model that is being used for these unfolded chains and just a Gaussian chain model is fairly close in predicting those shifts of about 0.4 units. There seem to be differences depending on local context that might be actually due to the fact that the IDP itself is not a Gaussian chain, but that one should really use ensembles of structures used from other experiment or derived from other experimental data. And that's something that we still need to revisit. But I'm sorry, and then you also asked about LLPS. Yeah, so this is something that is something that we're interested in. Yeah, there's a lot of charge-pi interactions and charge-charge interactions that are responsible for these phenomena. So yeah, and arginines, and it's not just that. I mean, arginines are not lysine, so there's it's an interesting area to follow. We're just making our first steps into that area, but yeah.
Thank you. Can I just ask, since you mentioned that arginine is not lysine, in what way for you is arginine not lysine?
I mean, it's not just the electrostatics. So the interactions, cation pi interactions are different, and arginine is a deal more hydrophobic, I guess.
Thank you.
Okay, there is a question from Marilyn.
So this is actually coming back a little to what Jim had just asked. So a long time ago, I published something which I think is underappreciated, which was that, as all of us sitting on this call probably could say that about something, that said that the backbone dipoles make the interior of proteins more positive inside. And that one of the key portions of that is from the interaction with its uronamide to each side, that because the hydrogen is smaller, the side chains always come off on the NH side of the dipole and not on the CO side. So, I mean, I thought at that time that the 0.4 shift of the ASPs in particular, which are very short, was actually not due to anything, you know, like you'd see that in a tripeptide. It's relative to a carboxylic acid. And so that's just kind of baked in from simply having two amides next to, doesn't matter what the conformation is. What do you think?
So if you look at the titration data for such an intrinsically disordered protein like alpha-synuclein, you really see that for the residues where there's no other charged groups in the vicinity, they tend to show ideal titration curves with a Hill parameter of 1 and also with the expected pKa model value. But if you go to the regions where there's alternating charges, you really see that right there, where you expect based on the electrostatics that they're shifted, they are also shifted. And in the case where it's alternating with positive charges, it shifts down. And in the region where it's mostly acidic, they all shift up.
And what's your model?
Oh,
So what's the model? Is it a tripeptide or a carboxylic acid, or what's the model?
So alpha. So in this case, we use alpha-synuclein, which is just an unfolded protein.
No, but I mean, when you're saying that the asps and glues are shifted, or they shift, what's the model that they're
Oh, it's the
shifted
model from Zhao. So it's the model. It's a Gaussian chain model, and the biochemical theory for the interactions between charges.
And one other quick question: do you ever see n smaller than 1?
Oh yeah, sure. Oh yeah. Yeah, so the both in, I mean, in the folded proteins, we see that all the time. I mean, n smaller than 1, right? Negative folded
Steeper, not shallower.
Oh, larger than 1.
Okay, well, I'm sorry, you're right, larger than 1. So steeper titration.
Not really. I think that there might be a few cases where, of course, if structural changes take place, then this could lead to such a positive cooperativity effect. But we have seen, I don't recall having seen anything that we would statistically be certain that that was a larger than 1, or it was just close to 1, but then the uncertainty was also large enough that we couldn't tell.
Yeah, it's because someone like Diane Barber, who sees pH dependence in signaling, they're getting these tiny, extremely sharp transitions, and I'm always on the lookout for any example, and they're very few. So thanks.
Yeah, not in this work.
Okay, there is a question from Adrian.
Yeah, so that was a great talk, Frans.
Thank you.
A question, tell me, so have you ever seen a deprotonated arginine? I know that you basically show that you tried and this one is protonated, but have you found any deprotonated ever?
No, I mean, the only thing we've been able to do is take arginine itself and dissolve it into super concentrated hydroxide, and then we were capable of making a titration like Lawrence McIntosh did. And I talked with no, I forgot his name. You know who I mean, from Johns Hopkins.
Bertrand.
Yeah, Bertrand. Yeah. So I talked to Bertrand, and he said that every time he tried to put an arginine into the center of a protein in order to try to get it deprotonated, the protein wouldn't fold, it would misfold, or it would generate swap dimers or other things. So I haven't seen one. They've been, I mean, this report is one, and there's another neutron report on other proteins that I know Lawrence has pointed out to me, where they also suggest this. So I don't know. I don't know enough about neutron diffraction and refinement of structures to be able to understand why this would happen or could happen, or whether it would be. I mean, we're left with a variance now between the solution structure is saying obviously that they're protonated, and the crystallographic data that is suggesting that it's neutral, and that has not been resolved. I would say, I think that probably the best thing that one could do is do solid state NMR, and generate the same protein in the same conditions, the same crystals, and then check what the NMR spectra would say.
Okay. And the other question is, thank you. Yeah, that's so. And then the other question is, when you did the unfolded state of GB1, right?
Mm-hmm.
This is unfolding with urea, or how do you make, or is it pH unfolded?
So Sarah Lindsay, she just produced the half proteins. So just the first half of the sequence and the second half of the sequence.
Okay.
And it's such a small protein it cannot fold. It will not fold into anything.
And then what kind of chemical shifts did you get versus reference for the unfolded, or they are all basically very close to the reference?
You mean in terms of the? Do you mean in terms of the pKa, or do you mean in terms of the chemical?
Yeah, the pKa, yeah.
I think those sequences were relatively acidic, so I would expect those to be slightly upshifted. But again, it depends on context. It depends on what the sequence is. But oftentimes, as also Professor Zhao has shown, the pKas in the unfolded state of several proteins actually match very well against this Gaussian chain model. If one is not concerned about a change of 0.1 unit, then
Right.
then these numbers are pretty good.
Okay.
We need to go into
Thank you.
experimental.
Now there is a question from Thomas Simonson.
Hello, everybody. Thanks, Frans, for a lovely talk. I have a question about the neutron diffraction data, which might be a little naive and I might be asking it sort of to the wrong person or people, but I believe the neutron diffraction depends on kind of fully deuterating the protein. And so is it sort of routinely successful to sort of fully deuterate the arginines when people do these neutron structures? I mean, I realize it's not your structure, but I just thought that there might be people in the audience who actually know a little bit about this.
Yeah, so I mean, of course we use for NMR spectroscopy, we do deuteration also quite often, and it is really possible to get very high deuteration levels for growth in E. coli, and also some other organisms that tolerate these high concentrations of D2O. But in this case, the positions that we're looking at are exchangeable positions. They're labile, so they basically are determined by the solvent. So in this case, it shouldn't be a problem to get them occupied to 100%.
Okay, thank you.
Okay, now Wei Yang has a question.
Yeah, I really enjoyed the talk, right? Indeed, actually, I spent a lot of time looking into this NMR experimental data, including old ones, including the classical experiment done on cell reduction. I think your talk today is much clearer in many aspects, right? Particularly one is pretty good. Actually, you show this apparent PK is sort of irrelevant to the intrinsic PK, right? This we really call this ionization cooperativity, right? I think there's some old data even in the cell reduction, right? It shows actually that might happen, but the data was not very clear, right? And so we look into that. I want to make a comment on this Huanxiang-Zhou's model. Huanxiang-Zhou's model on this Gaussian chain model from polymer theory, I think Candil was involved in this as well. And so this model is pretty limited, right? And Huanxiang show quite a bit of data actually they're relevant, but indeed actually we all know the unfolded state, there is a structure, right? It's molten global structure, right? And so some residues are still buried, right? I think that model is pretty limited, right? Okay, right now is my question, right? So my question actually, indeed actually, you mentioned actually how the chemical shift how the PK shift may happen, right? And so indeed actually, I think the field has really been looking into the ones which are very functionally relevant, right? Which may involve conformational coupling, right? Have you seen any of these? I mean, actually, when we will change the pH, actually, this the protein will change. Essentially, actually, we'll have conformation. Whenever you change the chemical state, and then there's conformational change happens, right? And then so the quite a bit signal will show up afterwards, and then
yeah. So in a lot of the things that I mean, everything that is involved with PGV1, those groups are all surface exposed, and so there, I guess the conformational states are not that important. I guess in the case of the photoactive yellow protein, we are in a situation that is maybe similar to what you described, where we have charges on two groups, or proton on one group and a charge on the other, and we can't actually titrate one group or the other, because as soon as we get to the extremes of the pH where we would modify one of them, the protein will change its state. It will either unfold or change its conformational state to the PB state, and so which is also known as the acid-induced state. So there, I think that there is definitely that's large-scale conformational changes. When it comes to conformational changes that at the level of the side chain residues, I guess these can occur inside more buried regions of proteins, but we haven't looked at any of that. And I think that it must have been Williamson who indeed for PGV1 suggested that some of the curves seem to suggest that there are conformational changes involved in the titrations as well. So that might be something to look back at. I don't know if
Yeah,
you
I
know.
have a suggestion. There's really some really good systems, right? And so like cell reduction, for instance, right? It was done very well before, but NMR technique was very limited, right? I think they did a really good job back then, right? I think this system has a lot of information in it, right? If somebody can really redo this experiment using the state-of-the-art NMR titration technique, there'll be a lot of details will show up. I think the problem in biophysics is actually whenever some system was done as classical system, we never revisit because out of fashion. If somebody can really do that, it'll be very, very useful to us.
Yeah. In that case, I mean, if you also are interested to follow the conformational states of side chains during titrations, I would also suggest to couple that then with the use of residual dipolar couplings, because originally NMR structures have been determined mainly by NOE-based methods, and those those structures are still not very high precision structures. And however, if you're interested in the orientation of individual side chains, then residual dipolar couplings are very sensitive to the orientation of individual side chains. So rather than trying to solve the whole structure, they give you very exquisitely sensitive data about the orientation of individual bonds in the structure. And that might be something that would be a good combination then.
I have a question. So let's say you have a protein that in its binding site has two tetrabond residues like two aspartate or glutamates. I have in mind the beta secretase, for example. And what can the state-of-the-art NMR experiments say about their protonation? Of course, they are close and they do interact. It's reasonable to think that they do interact. What is possible to say nowadays concerning their protonation state before and after binding the ligand that, in this case, beta secretase is binding?
Right. So I guess there are a few challenges there still. I mean, when we look at the basic side chains, it is quite easy to it's quite easy to detect whether these hydrogens are here because we can see the spin-spin coupling patterns. Also, Lawrence McIntosh has shown that for lysines, and Junji Iwahara uses this approach a lot, also for measuring the kinetics of these groups. When you go to the acidic side chains, it becomes actually more challenging because it is difficult to observe a splitting of this carbon with this hydrogen. Those couplings are too small to be generally resolved. So sometimes one can use isotope shifts by replacing hydrogen with deuterium, and that way the carbon chemical shift will change, and this is a way that you can detect that it's actually there. That's one thing that makes it challenging without a titration to see whether something is protonated or not protonated. But I guess isotope shifts are probably then the best way. The other thing is one about size. And there, I don't know what the limit would be nowadays for the size of a system, and because the experiments that we did, they relied on protonated. So I mean, protonated in this case meaning hydrogenated proteins, not deuterated. And in that sense, relaxation of these carbon coherences is generally quite bad. I think that you can definitely go and look at small enzyme systems of 30, 40 kilodalton would not be a problem at all. Of course, one has to do the assignment. I mean, one has to find out in the NMR spectrum where is which probe exactly located. But I guess the approaches that have been developed for looking at larger proteins by NMR spectroscopy, by deuteration and carbon observation, allow more complex systems to be looked at. And I know that Fleming Hansen at UCL is also doing that.
Okay, thank you. We have a question from Antonio Batista.
Okay, well, thank you, Frans, for the really great talk. This is I'm not really have a question related to what you did, but with something that you showed at the beginning. namely this titration curve with the up and down profile. And when I see some of these some examples like that, I always wonder if you ever observed a case like that, even after trying to disentangle all the NMR signals that like you are doing, whether anyone had observed a case like that in the protein, because I mean, I just know a few cases with small molecules like DTPA and stuff like that. Is this been observed in proteins? Do you know about it? Up and down patterns?
Okay. So when you say up and down patterns, it means that a certain site is for example, starting to titrate and then it becomes reprotonated, like you see in some of these tricarboxylic acids.
Yeah, exactly.
I have not seen a single case of this. And I think also that's because most of what we have observed, the side chains have enough distance from each other such that the coupling is not strong enough to observe it. And typically, even when we purify our proteins, we're not trying to make sure that they are free of any ions. I mean, we don't add any salt deliberately, but there might well be some buffer or some components from the protein purification that is there so that there is some ionic strength, and already some ionic strength will reduce these couplings, right? So I think if one wanted to observe them, I think one should have deliberately tried to make the coupling strength as strong as possible, like you can do in these example cases, which are really nice to explain how electrostatic coupling can work. They're beautiful examples. I know that there's very nice papers on that.
Okay, well, yeah, this was just more a curiosity of mine. Thank you.
Yeah, but I mean, it's good that you pointed out, because at least now the methods are such that when we follow the NMR chemical shifts, it really reports on the protonation state, so the potential at that particular site. So if these systems would exist, you would see them.
Yeah. Thank you.